Thank you very much for joining this webinar. We're starting a little bit late, so I will let Professor Sugai introduce himself. He's the WHO Collaborating Center on AMR for Japan, and we're very, very privileged to have a wide array of speakers today from South Africa, from Kinshasa, from Denmark, worldwide, and of course from Japan. And I'll let Professor Sugai go ahead. I've known Professor Sugai for a long time, and I really want to thank him very much for all his work he's been doing over the years on AMR. Professor Sugai, it's your turn.
Can you hear me?
Yes, we hear
Okay.
you very well.
Can you see the slide?
Yeah, we can see the first slide. We rely on you to click on, make your slide go down.
Okay, thank you. Thank you for inviting me to this AMR Pandemics webinar. This is Motoyuki Sugai. I'm director of the AMR Research Center in the National Institute of Infectious Disease, Tokyo. And today's topic about the trial of integrated One Health surveillance based on the WHO tricycle project. And the beginning, I'd like to introduce our AMR Research Center, which is established in April 2017, composed of 8 units: that molecular research, surveillance, and One Health research, field epidemiology, and MRSA and VRE, AMR genomics, and AMR fungus. And today's topic is about One Health, so I like to focus on the laboratory of One Health research in the group of the AMR Research Center, which is composed of 6 people. And so talking about the One Health research, it has been long discussed in the group of advisory group of the integrated surveillance of AMR in connection with the Department of Food Safety and Zoonosis in WHO. They, after a long discussion, they reached to the conclusion to develop the global harmonized protocol on integrated surveillance of ESBL producing E. coli in humans, the food chain, and the environment. That means 3 wheels. So it is called tricycle surveillance, which is epidemiological surveillance composed of 3 sectors, including humans and food chain and the environment, with the technique of the molecular biology. So I also joined the last ISAM meeting held, 8th meeting of the advisory group on integrated surveillance on antimicrobial resistance held in Utrecht in Netherlands in 2018. At that time, the protocol is almost concluded in the AJSA members. And here you can see the Miss Grants here, and myself is here. So all the almost all the experts of the AMR gathered in this meeting and concluded, and the result is the WHO tricycle protocol right here, global tricycle surveillance for 3 wheels for the human and the food chain and also the environment. And this is composed of the 8 working packages. The main package is composed of the surveillance in humans, and surveillance in the food chain, and surveillance in the environment. And also, this is linked with WHO Global AMR Surveillance System GLASS, and also required to use the information from the antimicrobial use and the antimicrobial consumption to discuss about the results of this integrated surveillance. And also the merit of this tricycle surveillance is this is a core tricycle unit surveillance, basic surveillance, but you can expand in collaboration with other extra surveillance systems such as surveillance for the CPE or surveillance for the other pathogens or for genome sequencing, and also the research for the residues of the antimicrobials in the environment, and etc. So I think this is a unique and very good opportunity for the new starter to start with One Health surveillance. This is a simple, very simple way to start the core surveillance system. And this system is composed, as I told you, human, food chain, and environment. And the human, there are two samples taken from the hospital's patient blood culture. For the community subject, the healthy pregnant woman was the original in the package, but we also added the food handler through samples. And for the food chain, the chicken fecal sample was taken. For the environment, liver and also animal slaughter wastewater, a human communal wastewater was taken. Sample were taken from these areas. And the very important point is this simple tricycle surveillance is composed of a single indicator, ESBL producing E. coli among the E. coli. So it's very simple, but very good for the beginners to start and get into the integrated surveillance of the One Health research. So in Japan, as soon as I come back from the last ISA meeting, I started to organize a team to start this One Health surveillance. And especially for us, the environment surveillance is the new to us. So we pick up several academia staff to collect the sample from the 5 prefectures over in Japan. And for the wastewater, we use a serial dilution method to dilute the sampling. And for the liver and the wastewater effluent, we use a concentration method, as listed here. By using this method, we establish our water sampling. And this is a simple results of the frequency rates of the ESBEC, which is a basic requirement to from this tricycle surveillance. For the human blood culture fecal sample, the ESBEC percentage is for the blood culture 30%, the fecal sample 12%, and for food handler less than 12%, 4%, and for the food chain, the Pote secret sample 2.5%. For the environment, liver is around this ratio, and for the wastewater effluent is 0, but for the human wastewater, around 3%. So the result tells that the proportion of the ESBC among the total E. coli bloodstream infection in hospitalized patients is as approximately 3%, whereas the proportion of ESBC among E. coli isolates from the stool sample for pregnant women was approximately 12%, so less than this. And for the food chain, the proportion of ESBC among E. coli isolates from the poultry fecal sample is about 0.7 to 3.8%. There are some fluctuation according to the company. And also the proportion of the ESBC among the total E. coli from human wastewater is, as I told you, around 3%. So this is the simple results, frequency rates of the ESBC from the tricycle surveillance. And also we counted the antimicrobial susceptibility, antimicrobial susceptibility, the resistant rates of the ESBC from each sectors. For the isolate from the human blood, like this is the radar chart of each antimicrobials. And this pattern is very similar to the isolate from the environment. But these patterns are quite different from the food chain. The food chain is much more susceptible to these antibiotics. So the pattern is quite different in Japan between the food chain versus human and the environment. We also did the whole sequencing of all the isolates from the ESBEC, which told us that the preference of the sequence type in each sectors. Here, in the blood culture, rectal swabs and rectal swabs was the prevalent human, rectal swab from the food handler, and also the water sample from the environment. The top hit is sequence type 131, which is a worldwide clone of the ESBAC, which you can see in not only the human but also the environmental sample, but not seen in the fecal sample from the chicken. For the chicken, sequence type 117 is the major type, which is also found in the number 5th of the sequence type in the water sample. So again, this tells us that the sample from the blood culture, human sample, and the environmental sample are quite different from the fecal sample in terms of the genetic characteristic of the ESBAC. And this is the genotype of the ESBLs in each sectors. Again, you can see in the blood cultures and rectal swab of the pregnant woman and the food handler, and also the water sample, one of the majority of the samples are CTXM27, which is again the most important CTXM type in the human infection. On the contrary, the fecal sample from the chicken is quite different. The CTXM14 is shared with other sectors, but another major ESB genotype is CTXM2, which is very rare in the other sectors, especially in the blood sample. So this is a simple result of the tricycle surveillance in Japan. So there are unique genotype and sequence type in the human and the food and environment. So respectively, for instance, ST131, CTXM7 is very predominant in the human and the environment, and also in the food, the ST117, ST2792, these are CTXM2 carrying E. coli, which is not rarely seen in the other human sectors or environmental sectors. So, but you can see in this common this type between the food and the environment is ST117 and CTXM2. On the other hand, the human and the environment common type of printed here, starting from the ST31, 38, 69. These are CTXM14, 15, and 27. These are the common type of the sequence type and the genotype. So in such a way, we can align the ESBEC isolates from the human and food and the environment. And the ESBEC from human had high genetic similarity to those from environment in terms of their STs, while similarity is food were very low. So this is the conclusion from the tricycle surveillance result in Japan right now. So in Japan, ESBEC from human hospital and community setting had high genetic similarities to those from the environment in terms of the sequence type and ESB genes, while similarity to food were very low. So this is a simple core tricycle surveillance result, but we are now extending this to more complicated genetic studies by using these isolates right now. And also, since we have accumulated with the technique of conducting tricycle surveillance in Japan, we are now trying to support the technical support of the starting the tricycle surveillance in other countries, especially in the Southeast Asian countries. The schedule like this that we send our staff to the collaborating country of the West Pacific region or Southeast Asia region, member countries, to how to conduct the data collection and analysis, support the technical support for the staff, and then after conducting the surveillance in the countries, they will send us the bacterial isolates or E. coli or the DNA of the E. coli and send back to our institute. And we have the high throughput DNA whole genome sequencing DNA system and running this whole genome sequencing high throughput factory, then isolate the data, and then with the data, we together with the member state staff, we are analyzing the data of the isolates to finish the first step of the core tricycle surveillance results. So we have already started several countries in Asia, and for the Philippines, we are supporting the technical issue in the environment. And for the Vietnam, we have already conducted the surveillance in Vietnam and isolated the sample, and the analysis of the genomic surveillance has already finished. And in Indonesia, also we have completed the isolation of the isolates, and now we are discussing about when to start sending the sample to Japan. And for the Malaysia, we have already received the isolates from the conducted surveillance in Malaysia, and we will have the meeting with Malaysian staff at the end of this year or the next year about the results, how to analyze, how to explain the results of the data from the Malaysia. And we are happy to start tricycle surveillance support in the United Arab Emirates next year. And we are very looking forward to collaborate with the people in the UAE. So this is some picture of the tricycle project. And this is a group of the tricycle in Indonesia. And also from the Malaysia, we also received the isolates. From the Indonesia, we also received the isolates. And also Vietnam. And now the tricycle surveillance in the Vietnam, organized by the Military Hospital 103 in Vietnam, was already finished, and we are now starting to write the paper with them. And for the Philippines, we conduct the support of to the Environmental Management Bureau of the Department of Natural Resources and Environment in Philippines in the environmental sector. And for the Indonesia, we are now conducting the surveillance together and also with Malaysia. So this is the simple explanation about what we were doing in the tricycle surveillance since after 2018. And also we are very eager to collaborate with other countries who are willing to start tricycle surveillance because tricycle surveillance is quite So we link with graphs. So all the data comparison is a very important issue from the point of the worldwide. So thank you very much for the listening.
Professor Sugai, we thank you very, very much. I remember when my friend Antoine sent in this to announce the tricycle proposal in Agiza, and the photo you showed I was in was very, very, very interesting. I noted that you said you started in the UAE.
Yes.
Can you say a few words about that?
Yes, because this tricycle surveillance is supported by the Japanese government, especially in the this Aspire program. And we have discussed about issue with Ministry of Health, and they said yes to start collaboration with United Arab Emirates, because it's not in the Southeast Asia, but they are willing to support. So we are now one of the member in the University of the United Emirates Arab is coming from the he moved from the NIID to United Arab Emirates last year. So we are in close collaboration, and we are now preparing for the start of the tricycle surveillance next year.
That's very good. So, as you know, since we are proposing our in-person, we have had to postpone our in-person conferences. Well, make sure that you were with us as a speaker, and we have the Emirati authorities here too. And I think that the tricycle project is interesting. It's an illustration of the coordination in terms of One Health, in which we see that the animal sector is interlinked with the environment, interlinked with the human beings, and it's actually, if we're serious about AMR, we have to tackle the problem in a comprehensive way, in a really One Health way. We try to organize this webinar today with that concept of approaching the situation of AMR from different angles: water, wastewaters, human health, and animal health. And each of the speakers is developing a different aspect of this. Dr. Tuzi, do you have any questions, comments?
Yeah, I was fascinated by the excellent presentation by Professor Sugai. I mean, it's and also your comments. I think this is after the first webinar earlier this morning. This is continues that theme, and I think we definitely need to develop it further. It's a great start, and I think we should really develop it further. I don't have any specific comments, but I think it highlights the importance, I think, of this topic and developing it further. I think Garrence's initiative, if you will, with the expert health team, expert One Health team, and all the global experts together. I think it's great to get them together initially in webinar settings, and then having established that link by then making a really decent conference where all of these items can be not only presented but discussed, and have an interactive discussion on the ways forward, because it's a complex topic. I mean, we established from this morning, it's not easy to do these surveillance. It's not easy to implement. It's not going to be easy. It's one of those things that's not going to be easy, but it's very important. So I think we need to have this discussion to make sure this happens. Thank you.
Next speaker is Professor Luther King Abia Akebe from South Africa, where we have projects related to water and wastewater. It's in the South African press over the past 2-3 months, there has been articles on dramatic issues with water shortages, water leakages. It shows that the entirety of the water and wastewater system needs to be rethought. So it's really a priority country and a priority example of very high level experts in the country, and at the same time a very dear situation that needs remedial. So
Yeah, just 2 minutes. Luther King,
2 minutes for Professor Luther King to come on. We had met at the EDAR, which is the yearly or bi-yearly environmental determinants of antibiotic resistance conferences that have taken place in a number of countries. This year it was in Montreal. Last year it was in Sweden, and it had been in Hong Kong in 2019 before COVID-19. And it was very important because actually the EDA was initiated in Quebec with about 15 people some years ago. So, I mean, their story really moves me because it's so that what we're trying to do now with our prevention, preparedness, and response approach to AMR and pandemic. In other words, to try to deal with topics that not everybody is aware of. Try to handle it from the standpoint of prevention. And we're waiting for connection with Luther King. And Motoyuki Sugai, you did, I mean, it's really fabulous work, this work on the tricycle. I'm very glad that it's coming to the UAE. Huh? Dr. Tuzi, what do you think? It's good it's coming to the United Arab Emirates now.
I think the fact that it's coming to UAE and the collaboration between the national bodies in places like Japan and the UAE is a very hopeful thing, and definitely we would like to, as an educational arm or educational organization, we'd like to be involved in assisting where we can in getting everybody together to have that discussion. So if we, definitely we can have a role in that, that would be really important because we would love to facilitate all of this. Do you have anything to add, Professor Sugai? I mean, do you want to add any comments?
Yeah, at the moment, we are now expanding the tricycle surveillance, not only the E. coli, but also expanding to gram-positive and also other type of the resistant bacteria. But the initial step of the starting the tricycle is still, I think, the E. coli is the best choice. And also the comparative study between the country is very important, I think, especially in the country which has a close relation of the E. coli between the human sector and the food sector, and also the food sector and the environmental sector. We need to carefully analyze the common E. coli in these between sectors, and which will tell us what type of the plasmid or what type of the resistance gene is really circulating in the country. And this will be the one of the topic that we can start conducting the more robust surveillance in the country. So I think this tricycle surveillance is a very good start point of the more comprehensive, large-scale surveillance in the country. That's my comment.
Thank you very much. I have there's a question in the previous webinar, which I'll put to you because you're equal expert on, especially with E. coli and stuff, and transmission of genetically transmission of resistance. Are there any solutions, or is it once we specific solutions? I mean, I know we need to use narrower. We need to have better stewardship programs, and identify antibiograms and have much more comprehensive work. But are there solutions to prevent the genetic transmission of resistance between different species?
I think it's a very difficult question, since, for instance, poultry, in tricycle surveillance, we are using the isolates from the chicken. So, but the chicken farming is everywhere in the country, but the style is completely different according to the country. Some country are using the family organized poultry production. Some countries are using the more robust factory organized production of the poultry, chicken meat. So it depends on the country that how the food processing system is prepared, and we have to see the link, how the link between the food system and the human sector, the food system and environmental sector. And this could be the second target after obtaining data for your tricycle surveillance, and then what we can do for the human side, what we can do for the food producing sector, and how we can grasp this relation, or how to cut the relation in such a way. We have to do the brainstorming with the government, how to control the spread of this antimicrobial resistant bacteria. So it's very complex, but it's not so easy, I think.
I understand. And that's why I think it's important to have these education and these seminars and discussions, because it's the easy topics, okay, we can deal with, but the difficult ones, people, it's difficult to deal with them. And until we have the expert people together and really discuss simple ways of moving things forward, we will not make a big difference. So I think that's why we appreciate you being here, and also Professor Yamagishi earlier in the morning. So I think having this global discussion with experts is going to be really important going forward. Thank you.
Thank you very much, Professor Sugai. I hope you can stay with us. We now have Professor Luther King, Abia, Akebe.
I'm going to be talking about antimicrobial resistance, One Health, research evidence from South Africa. If we look at the brief history of antimicrobial resistance or antibiotics, we will realize that when antibiotics were first discovered, they were considered a magic bullet. So they were used to treat every single infection that human beings they have at any time. And because of that, especially during the wars, a lot of people could go back home to their kids, to their families, because there was a treatment for infections that they were getting. This Eureka moment for humanity has been regarded as one of the biggest discoveries of humankind. However, this discovery was very short-lived. For as soon as antibiotics were discovered, their use skyrocketed, and they were used in animals, humans, in the environment, and this increased use and indiscriminate use of these chemotherapeutic agents led to an increased resistance to AMR almost immediately. So, what this resulted in is placing antimicrobial resistance at the comparative level to other pandemics that have existed. So if we look at the history of pandemics in the world, and we look at the three major pandemics, for example, that have been recognized as one of the greatest cost to human life, we'll look at the Spanish flu, the HIV, and the recent COVID-19. But when we look at AMR, and we place AMR just in 2019, we realize that just in that year, in a single year, AMR claimed over 6 million lives. This means that if we were to compare AMR, and if we continue, or if the rate continues at the way it is, it means that we will end up with more deaths than any of the pandemics that have ever existed. And this therefore translates, if we look at the ONU report, this translates to about 10 million people dying by 2050, costing the world hundreds of trillions of dollars to the world economy if nothing is done. And while we are looking at this 2050, which is just around the corner, some other regions of the world are even foreseeing that by 2030, the situation will be worse. Can we therefore stop the use of antibiotics? Will that be a solution? I don't think this is possible, because I am very sure that many of us must have read below the line, meaning that it's not possible to say don't use antibiotics or use antimicrobials. That's why many world organizations or renowned organizations have come up with the whole concept of the One Health approach, which advocates the intercommunication between the human, animal, and environmental sectors. And when I talk about the environmental sectors, this includes plants, because it is only through this communication that we can achieve an effective fight against AMR. Within the antimicrobial resistance unit of the University of KwaZulu-Natal, therefore, we developed this framework that will ensure that we carry out surveillance of humans, animals, and the environment to determine the burden of AMR within South Africa, and so that we can therefore suggest interventions ranging from biomedical right down to policy that will help curb the situation. And we carried out the studies in one of the districts of KwaZulu-Natal, called the district, whereby there are many hospitals, major wastewater treatment plants, major animal farms. What we did in humans in this sector is that we collected E. coli from humans using different surveillance approaches, and we subjected this one to a panel of antibiotics. And what we found is that there were varying levels of resistance, or varying percentages of resistance on all the antibiotics that we tested. When we did the same work in the same district, in one of the biggest poultry producing entity of the district, we found that there was resistance to different microorganisms, and we used the farm fork approach, meaning that we followed the chicks from the time they were hatched until they went to the abattoir and ready for the table. And we found antibiotic resistance continuum and different organisms, including E. coli, Enterococcus, Salmonella, Campylobacter, and others. We did a similar study using the pigs and going through the same farm to fork approach. And we also found different resistance patterns and at different levels at all the sectors of the continuum. In water, we found wastewater treatment plants within the same district, and we found different levels of resistance, including high resistance to extended spectrum beta-lactams due to extended spectrum beta-lactamase producing E. coli, which were our target organisms. So we therefore proceeded to select multi-drug resistant isolates from our panel of isolates from the three sectors, and we subjected them to whole genome sequencing and compared their genomes using phylogenomics. What we found is that there were clustering isolates from the various sectors, showing that there was resistance between these three sectors. Did the same thing with soil samples that were collected from manure amended soils. And what did we realize? We also saw that when we expose or when we analyze the isolates from the soil that was amended to the soil that was unamended, together with water and human isolates, we also found that there was a connection between the amended soil and the unamended soil, which means that there was a transfer of microorganisms or resistance from the manure onto the unamended soil. The same thing, we realized that the litter that was used to fertilize the farms contains some isolates that were unique, meaning that the litter was a source of antimicrobial resistance due probably to the use of antibiotics in the farms or promoting chemicals that trigger antibiotic resistance. And the same applies when we now further look and we phylogenetically isolate existing databases. And we found that there was also a connection or clustering of our soil, amended soil isolates together with one isolates. What does this tell us therefore? Therefore, it tells us that there is a complete movement, or there is a movement, there is adequate movement of antimicrobial resistance between the three sectors. And just as we look at the video that goes on there, if we consider our fellow, rather they as antimicrobial resistance or disease pattern, which is not the case, please. I think this is just for the case of the production of the presentation. We will realize that he couldn't set up a single stick. But by the time he combined the three sticks, they decline on them. This is just a summary to say how to achieve a universal fight against AMR. Then we need all the three sectors and we need adequate communication. Because as of now, we still have doubt or we still have lack of information on the effective contribution of the environment. For example, we understand the human health sector very well because a lot of financing has been placed in there. We understand the animal sector very well because a lot of financing has been in there. But the environmental sector still needs that same attention so that the One Health concept can be fully applied. Because that's the only way we can achieve fight against AMR. I would like to acknowledge the following persons who I presented their papers there. I have antimicrobial research. I would like to thank for your kind attention and the room is open. If you have any questions, I'll be very happy to respond.
You guys get up and down and then they see you walk past.
Do we have a question there? Thank you. I have a bit of connection problem. In fact, a serious connection problem. So that is why we had to have the recording. But I think after the presentation, if there are questions, I'm very welcome to take them up. I am Luther King, and I am an applied and environmental microbiologist at the University of KwaZulu-Natal. Although maybe if I didn't introduce myself at the beginning. Okay.
So Luther, tell us a little bit about the situation. Tell us more about the situation in South Africa. How do you see it going? I mean, you guys, you've had a lot of water problems, like I said, over the past few months, and of course in the past too. And how do you see the future in terms of the people concerned with the AMR aware of the role of?
I think we are going on and off, but yes, you were asking about the situation in South Africa. Did I get you clearly, Garant?
Yeah.
So please ask your question again. I didn't get it clearly because it went off.
People working on AMR nationwide, in terms of your government, aware of the importance of water and wastewater in AMR control, or are they focusing only on people taking their pills properly? I'm being provocative, but a lot of countries have that problem.
Yeah, that is a problem, a general problem with many of the African countries that are visited. However, South Africa is doing some works and doing a lot of work on AMR. It is also in the National Action Plan of the country, which is now expired this year and needs to be revised. So I think a new version will be coming out. But AMR is regarded as one of the major health issues within the country. So yes, research is being carried out. However, maybe in my next presentation, when I'll be talking about wastewater, you will realize that although that research is going on, it's not equally distributed across all the provinces of the country. So the research is focused just in some of the provinces, like the KwaZulu-Natal and some other few ones, while other provinces are just not carrying any research in that field. And I think it's also because I think research capacity is one challenge, and then the technical expertise is also challenging for some other regions compared to some other ones. So, but all in all, there is research that is going on in South Africa, and just as I presented on the One Health concepts, it is gradually gaining grounds, and the environmental components is also coming in, though still at snail pace, but it's coming in. Yeah.
It's ironical because Professor Sabiha Isaac is a world famous AMR researcher and spokesperson, speaking at every ministerial meeting and global health meeting, and she's South African, and she's really been a leader worldwide in the AMR awareness, as you and I know her very well. And so it's a bit ironical that actually people in governments don't understand sometimes how AMR works and how it is a very global, One Health issue. I know that's the case in France. It's even the case in the US. So, you're South African are in good company because it is a global problem, and a global response is still somewhat lacking in spite of ministerial meetings. Dr. Zaman works with the International Center for AMR Solutions, which is trying to do something very original. I was mentioning Sabiha Isaac before. She's on the board of ICARS. And ICARS is trying to support countries in implementing solutions as opposed to just watching the situation, but rather trying to remedy a bad situation that we have in many countries. And in terms of the initial program, Didier Bompangue will intervene after Dr. Zaman because Dr. Zaman has urgent matters to deal with this morning. So Dr. Zaman, it's for you.
My name is Wesal Zaman. I'm a social and behavioral science advisor at the International Center for Antimicrobial Resistance. I've been working with ICARS for just more than 1.5 years right now. ICARS, the International Center for Antimicrobial Resistance, is based in Copenhagen, and we have a very diverse team here. We right now we have expertise in all across One Health, from human, animal health to environmental health as well here in Copenhagen. So what is, I just will give you some introduction to ICARS, and then I'll go into some of our projects that we have that we have funded from ICARS side. So ICARS is an independent and self-governing institution that's working internationally in partnership with low and middle income countries and governments. So mainly we work with governments and also research institutions. I will say a bit more about that later on, how we work on that. So what we do at ICARS is we provide funding, but we also provide expertise and expert advice across the One Health spectrum. That is the uniqueness of ICARS, where we do not only fund projects, but also support them scientifically throughout the project development and also project implementation as well. ICARS was initiated in 2018 and by Danish government, but right now it is a self-governing institution. We have over 40 employees here now in Copenhagen, but our funded projects are across the world. So what we do is we work to reduce drug-resistant infections in low-middle income countries where there is a need for it. The way we work is that our projects are supposed to be evidence-based. We really focus on evidence-based projects. They are context-specific, cost-effectiveness, sustainable, and they cover across the One Health spectrum. By covering across the One Health spectrum, it doesn't mean that all projects should be One Health, but we also fund projects that are only animal health, human health, or separately environmental health, but the organization overall works across the One Health spectrum. So our mission is to partner with LMIC ministries and research institutions to co-develop and test tailor-made solutions with potential for scale-up across the One Health spectrum. So the way we work is we work through intervention and implementation research. We co-develop projects with our country teams, and when the implementation is done, the final idea is to provide a proof of concept to the government to upscale the project. For these projects to happen, it's for the sustainability of the projects to continue. There's a need for a political will. That's why we work closely with governments. We value ownership throughout the whole stakeholder chain. This means that when we develop projects with country teams, we engage in a long-term co-development process where proposals are developed with the country teams, and they engage different stakeholders from within that country who are relevant for that AMR problem that exactly that project is trying to tackle. We also want these projects to be context specific, meaning that we understand that every project, every context, every country, and every AMR problem in a specific context is unique. That's why we value this context specific work that we are doing. We understand that one solution is not for all countries. That's why we work closely with all of the countries to just develop solutions that are specific to their own context. The way we work is a unique approach. As I mentioned before, we engage with the governments to see their problems. These governments are the ones who set up priorities for the AMR. So we really value their work. We also require a commitment from the high political level. And then we also work with the researchers and institutions in the country. So we bring these two top-down and bottom-up approach together to foster collaboration to effectively tackle AMR in LMICs. ICAR's core principles are country ownership. We really value ownership of these projects. You can go down slide number 6 right now. Yes, here. Thank you so much. So the core ownership is we really value that the projects are executed by local researchers. That is, the priorities is based on local needs and challenges. We work across One Health. We try to collaborate with different sectors, partnerships. We deliver evidence in partnership with key stakeholders in the country, but we also try and work with other international stakeholders in the countries that the projects are being implemented. All our projects are solution focused, where they can produce scalable evidence-based solution for policy, programs, and practice. So we really take these proof of concepts. All of ICAR's projects, they have 3 different components. One component is policy, one component is behavioral change, and one component is economic aspects. So we want to take all these 3 different aspects and create a proof of concept to give to the governments to show that these projects work or whether they don't work. So normally, ICAR's funded projects are they run for 3 years. Overall, it's an implementation where the project, when developed, we try. Like there's a lot of project going on around the world where there's surveillance, data collection, but we want to apply work in the context. We try to give solutions and then see if they work or not. We work across these different pillars, as you can see, 5 pillars. You can go to the next slide. I won't go into details about these pillars. You can read these pillars a bit more on our website. Currently, ICARS has 41 projects in 21 countries and territories. As you can see, our projects are in South America, in Africa, also in Asia, and in parts of Eastern Europe as well. You can see that they are across human, animal, and environmental health. So we work across all of them. So we have 3 different types of projects. We have ICARS's only funded projects, which is co-developed between ICARS and LMICs. We have projects that are co-funded. They are developed with other funders together. That we fund, and some of our environmental projects are through the JPI AMR project that we had, and it's still continuing. You can go to next slides. I'll go through these 2 very quickly. So ICARS has a board of directors. There is also a technical advisory forum. Next slide, please. So the technical advisory forum is prominent researchers from around the globe. So whatever proposal the countries develop, it is also seen by these experts in the field, where they provide feedback. So there's a lot of, as you can see in throughout the development of our projects, a lot of stakeholders are involved, and that's what it makes it unique, so that it's also solution focused, it's context, but there is also a lot of expert knowledge from outside the country as well to help and support that. If you can go to the next slide. Within the ICARS's science team, as you can see in the next slide, there is a range of advisors that work with the country teams. We also have consultants. Grant, you mentioned a few minutes ago, Dr. Sabiha Isak. She's also one of our consultants advising us, engaged in ICARS's project developments. She actually oversees a lot of our projects that is proposed for funding. So you can see there's a wide range of expertise from ICARS that we try to help our country teams. Previously, ICARS since we're talking a lot about environment, most of our projects right now are human and animal. We have only a few environmental projects. That's why in the mid of this year, ICARS requested for proposals specific to environment. And now we have a range of proposals that have come, and hopefully these projects will start being implemented in the coming year. So I'll just quickly go through a few of these projects that we're doing right now and funding. You can go to the next slide, next slide, the hot mask project. So one of the projects that ICARS funded through in collaboration with JPIMR is the removal of AMR genes and bacteria from wastewater using modular advanced treatment solutions. This project is done in Ghana. It is in collaboration with the University of Ghana, and also there are also other external partners to this project. The Karlsruhe Institute of Technology in Germany, Norwegian Institute of Water Research in Norway, and University of Nairobi, Kenya are involved in this project as well. So they are trying this new technology, the blue light technology, to treat wastewater from abattoirs and farms and hotspots of where waste is being disposed into the environment. Currently, the project is a bit delayed because this blue light technology was really difficult to achieve, but it is now in Ghana, and they're testing it currently as we are speaking. So the other project that we have funded is the mitigating the spread of antimicrobials and resistant microbes through the treatment of manure in Tanzania. So the idea here is that poultry manure is a desirable fertilizer used in crop production, aquaculture, and other food productions. The use of manure could lead to increased environmental contamination with antimicrobial residues and antimicrobial resistant pathogens and genes across the food chain. So as you can see, there's a bit of a One Health connection here, where we work with poultry in the poultry section, or we work with manure, but also the idea is to prevent it from contaminating the environment. Next slide, please. The next project that we are co-developing currently with Zimbabwe. Right now, it's in progress. I did a scoping visit there in the beginning of this year. Is the strengthening the environment regulatory framework for AMR in Zimbabwe to reduce use of antibiotics. This is a very interesting project. If you go to the next slide, I won't read all of this. This is just the context in Zimbabwe that the problem exists. But in the next slide, you can see what they are really trying to address here. One more slide, please, to this figure. Okay, for some reason you cannot see the figure in that slide, but what they are doing is trying to develop standards and regulations that would capacitate the regulatory agencies to improve enforcement. So in Zimbabwe, there is, for the amount of waste that you dispose into environment, there is a tax that you need to pay. So they are trying to add this: if an abattoir or a poultry association tries to treat the waste with antimicrobials, then they will pay less tax. This is just to sensitize them into doing this action. So this is one of the interventions they're proposing. The other one is in Zimbabwe, they have different types of abattoirs that have, some of them have huge lands capacities, others are in the city that do not have a lot of a lot of land. So in these projects, they are trying to treat the wastewater before it goes into environment in different manners in the abattoirs that are poultry farms that they have a lot of land. They are suggesting an anaerobic facultative maturation, which is through natural UV attenuation for abattoirs with land capacity. So this is, they try to do these natural treatment of antibiotics in different ponds. But if they do not have the land, then they're using if they're using septic tanks, they use UV treatment modules. And on the other hand, since these are bundle interventions, they're also proposing to educate and aware the proper use and disposal of antimicrobials for these abattoirs. The next project that ICARS has funded is decreasing the carbapenem resistant, the CRE transmission between hospitals, communities, and domestic animals. This is also, as you can see, a One Health project overall. So most of the projects that I've just presented to you, you can read more about each and every one of them on our website in details. It's a lot more detailed than this. There is a scientific team in country that is working on this, and 2 advisors from ICARS on each and every one of these projects that we do. And as I mentioned, these were the 3 different projects that we are going. ICARS is fairly new. It is only 4 years that we are here. So most of our projects do not have their final results, but we can see that we lacked a bit of environmental work in our profile. So now we have added a new RFP where we have way more projects looking at the AMR in environment. So that is just a quick presentation, and thank you so much for listening, and I apologize for all the technical delays. If you have any questions, you can ask me now. Thanks a lot. You can unshare the slides. Yeah, can anyone hear me?
Yeah, we can hear you, Dr.
Great.
Thank you for that.
Yeah, we can hear you. We're trying to look at the questions, at the
Yes.
Q&A.
So I can see one question that says, I'm an AMR researcher in Nigeria. How can I join ICARS and how can I fund AMR projects in Nigeria? So we work with LMICs, but there are some countries that is difficult security-wise. So, but still, if you are interested, you can submit an expression of interest to ICARS. You can go on our website and check. There are 3 different deadlines throughout the year that you can submit an expression of interest. This expression of interest is only very short, 1 page that talks about the AMR problem in the country and the interventions that you're proposing. We look at that expression of interest from different countries, and if we see that there is potential in working with that, then we come for a scoping visit to your country. We work with different stakeholders and develop a proposal with you. So you're welcome to look at our website and find there's all information on the EOIs. Expression of interest is on the website. You can check that. Thank you.
And these, just as a question, so these projects, at the moment you focused on Africa, but you can, I guess they have a global scope?
The projects are in low and middle income countries, so it's Africa, South America, Asia, and parts of Eastern Europe as well. So that's where the projects are. Many of our projects currently are in Africa and Asia, but we are also getting a lot of good projects from South America as well. So yeah, it is global, but within the low and middle income country context. We do not fund projects in high income countries because they already have the potential, so they can fund their projects themselves.
Are you looking at efficacy? I mean, is part of the thing to look at the efficacy of these projects so that they can be rolled out globally? I mean, is that part of the remit?
We're looking at to roll out a project globally because we think all projects are because context is really important to us. We do look across the fidelity of the projects, the effectiveness of it within those contexts, and sustainability of it, and scale up possibilities of it within that context of the country. So we are not trying to look at efficacy for global purposes right now. I have another question on the chat. It says, how do you do monitoring after funded a project? We have different methods for monitoring these funded projects. Since 2 advisors from ICARS is involved in this project, we have very regular meetings with our project teams. We do country visits, and we have a monitoring and evaluation team where they have to report every project that has submitted or funded. They would have to submit quarterly, biannually reports depending on the project type, and then we carry out those monitorings as well. So there are different ways that we monitor through regular engagement and also through regular monitoring phases that we do. It's okay if people have other questions, you can always send an email. If you go on our website, you can find my name there, an email address as well, and just send me an email, and whatever questions you have, I'd be happy to support you.
Okay, so I thank you very much. I know ICARS, I remember first Zoom with ICARS and the head of your scientific team, and you emphasized that you are not really going into surveillance in the sense of not just watching, but you want to bring solutions. And that's, I think, a remarkable initiative. And you've grown a lot since I've known you, learned about you from Sabiha Isaac, that was like 3 years ago. You've really progressed, and it's a good omen for AMR control. We thank you very much for intervening.
Thank you so much. Thanks.
Good morning, everyone. Thank you very much for having me in this webinar. My topic I'm going to share with you is talking about the challenge for creating the One Health Institute for Africa at the University of Kinshasa in the Democratic Republic of Congo. I'm Didier Bompangue. I'm Deputy Director of One Health Institute for Africa in DRC. This new institute is managed by the Professor Jean-Jacques Muyembe, who is director. So DRC is one of the biggest countries in Central Africa, with more than 120 million of population right now, with a very fast growth of population. So this country is one of the most affected by complex crises, talking about economic, social, political, and also climatic crisis. So all this context is also in this context is developing a lot of emerging diseases. For example, cholera, Ebola, Mpox, chikungunya, salmonellosis, tuberculosis, and also malaria. So one of the well-known right now is the Mpox outbreak. Since 40 years, population in DRC is dealing with this very neglected disease. And the good news is since maybe 4 years, the Mpox change is not anymore a neglected disease. It's become a very well-known disease with a lot of focus of international concern. But as you can see in this slide, this disease is affecting DRC since many years, and all the country is affected. And the most important burden of disease is the zoonotic, the zoonotic aspect of the disease. But till now, we are struggling to have the good understanding of the dynamic of the disease, of the model of transmission inside the population, and also for treatment, there is a lot of challenge. The second example I would like to share with you is about cholera. So the cholera is one of the very important outbreak in DRC. DRC is one of the most affected country in the world. There is since 15 years cholera national plan in the DRC, but we continue to struggling with the cross-border management of this disease, and also some occurrence of antibiotic resistance for using some drugs. Of fresh therapy could be one of the good options for fighting the disease, but we're still struggling in the global understanding of the disease. The last one example is about the Ebola outbreak. And right now, there is a lot of challenge, for example, environmental reservoir, how the outbreak is starting from survival. This is the new, one of the new information we have in Ebola. So the outbreak is not starting only from a zoonotic aspect, but also for survival. So how to manage this kind of situation? People, the country still struggling with this. And lastly, we are going also to talk about the burden of bacterial antimicrobial biology resistance in DRC. As you can see, we know that DRC is one of the most affected country, but at the same time, the country is also one of where there is less information. So with a very few information, the country is appearing like one of the most affected. So I think this is a very major threat the country is facing. So to try to support the country in the future to deal with all these major threats, public health, one of the ideas we have in DRC is to focus on implementation of One Health approach. So this idea, we are working on that since 10 years with in University of Kinshasa, mainly in the Faculty of Medicine. But last year, we worked on implementation of the One Health Institute after 10 years of working in training people with in the Master of Ecology of Infectious Disease. And right now, with the idea is to move to the One Health Regional Doctoral School in order to strengthen the capacity of the people. So that One Health Institute for Africa, we are looking forward to implement this because there is the mismatch between the training of and the healthcare need. And there are also inadequacy accumulated over the last 10, 20 years in this context we presented before. So we need the diversification of the profile and the skill of human resources and the health problem in order to face the problem we are facing in social and ecosystem. And for all this, I think the One Health approach is the most important. And so the One Health Institute for Africa, we are setting up in DRC, will be working in research, capacity building, to support also operational activities. And also we are going to work for setting up the observatory of health of social ecosystem. So because of, and for that, we work in last May, we organized in DRC the international workshop with many many countries and many partners in order to figure out how we can move forward with this idea of setting up One Health Institute in DRC. And so the main conclusion of this workshop it was to try to externalize this international One Health conference in these low-income countries, because we know that there is some international workshop for One Health, but for these specific countries with very less resources, it's very important to focus on this country. The specific workshop in this country will be very important. That's why we are advocating for more finance to strengthen and to this One Health approach in these countries, and we are expecting to have the next workshop on One Health in this country in 2 years. And finally, for the future, the idea is to focus on setting up this One Health regional doctoral school to train people from the country, for many African countries. For this regional doctoral school, we are thinking to start in Kinshasa. It's not just one initiative for DRC, but this is an initiative for also more than 10 countries in Africa. We have colleagues from Morocco, from Tunisia, from Senegal, for Benin, for Cote d'Ivoire, for Burkina Faso, Cameroon, Congo, Brazzaville, and DRC. And also for the time being, we have two teams from Europe, and for Belgium, and also for France, the University of Franche-Comté of Besançon. So we still very open because we think that training, supporting the capacity building of the people in the field where the problem is occurring. This is one of the very good strategy to be ordered globally to figure out these major threats, starting in these very remote places and affecting all the world. So because of that, we think that it will be very important to integrate this One Health Institute in Kinshasa in the global network of working on One Health. That's why we would like really to thank Professor Garance for inviting us to join this webinar. And we are looking forward to hear from you and also to receive more support from you to strengthen this One Health Institute in Kinshasa, where there is many problems. And I think as the world, as the global response in the world, maybe should start where the problem is occurring. That's why we receive the support from all of you to strengthen this regional doctoral school and also this international One Health Institute in Kinshasa. Thank you very much, and over to you.
Thank you very much, Didier. That was very good, and I was very happy to be asked to be on the board of your One Health Institute, where, I proposed that I'll do some advocacy for it. And we're just starting this. And I think that immediately over the next month, maybe we can do the same webinar, being better prepared on One Health in Africa in French, because Luther King speaks French, you speak French, Pote, and myself, that's 4 of us. And I used to work in Francophone Africa, and I know just most people working on AMR in French-speaking Africa. So we should do a French webinar coming up next month with also the people who went to Kinshasa for the launch, which I was not able to do. Thank you very much. That was very good. John Pote and I have known each other for a number of years, and we did together webinars for the WAAW, the World AMR Awareness Week of the WHO. We did 2 of these in Abia. We're waiting for the engineer to put up your presentation, John. So I'm saying a few words. And John, I think he will introduce himself, but he's worked in Cuba, he's worked in India, he's worked in many African countries. He's also a professor at Kinshasa University, and he's really very well known also for his work in Switzerland, and he's head of the microbiology lab in the University of Geneva, and he's become Swiss. He's Swiss Congolese, and he's sort of an international figure now on One Health and the battle against AMR. So we are very, very happy to have my friend John come on. The webinars and seminar we did 2 years ago. We had the scientific lead of ICARS. You heard Zaman before. We had Vivian, the chief of ICARS, in 2022. We also had Virginia Tech. We had John Allen from Harvard University. And of course, John, we had the Swiss authorities for a day-long seminar, which was on the web. It's still on the web. Unfortunately, we had simultaneous translation, and the translation is not on the web. So the morning is in French and the afternoon tends to be in English. Now, as soon as you're ready.
Okay, I am a head of environmental microbiology group at the University of Geneva. I am working in many countries, mainly according to the degree of development of a country and the climate. I work in Switzerland, in Cuba, in Morocco now, and Sub-Saharan Africa, mainly in Congo and Senegal. For the objective that I am doing mainly, it's to provide an overview approach for the classifying and characterize antibiotic resistance bacteria and antibiotic resistance genes in wastewater according to the degree of development of country and the climatic condition. The main approach are focused on assessment and the prevention of pathogens, antimicrobial resistance, and we identify many sources, and we see what problem if we compare the climate conditions, Switzerland, India, Morocco, Cuba, and the Congo. And for that is to develop the approach for the detection, the quantification, characterization of FIB, antibiotic resistance bacteria, antibiotic resistant genes, and to see what approach for the wastewater treatment to limit the spread of bacteria and resistance genes in aquatic environment and the food chain. Okay. And
So
for the methodological approach, there you see, we are doing the detection, the quantification.
We're on slide 2. That's what you want, research focused, problematic, methodological approaches.
Yes, okay. Another slide, yeah. Next, please.
Okay.
Next slides, yeah.
Yeah, okay.
And if you see our conceptualization and the modeling study, here we have developing countries and we have developing countries. You see well, for the developing countries, there is no wastewater treatment plant. It goes donc all the wastewater go directly to the receiving system. And in the developing countries, you see that there we have the wastewater treatment, and the wastewater treatment are not optimized. And there we have problems in the river receiving system, even we have the wastewater treatment plant. And there we are doing for the CRE and the ESBL, E. coli, Pseudomonas, and the other. And the 5 main questions that we are doing for our research is to ask, firstly, what is the origin and the role of pathogens, antibiotic and antibiotic resistance bacteria, and the resistance genes in aquatic environment with respect with wastewater treatment according to the degree of development of a country. Secondly, what is the role of a climate? If you have the pathogens, if you have antibiotic resistance bacteria and the genes, and their accumulation to vegetable, to animals, to fish, and what is the problem for the nosocomial epidemic in the hospital. And therefore, the interdisciplinary approach, we see there is many physical-chemical parameters, such as the presence of many pollutants, micropollutants, that has the problem and increase the antibiotic resistance exchange. And we are proposing the solution. What can be the solution for all of these approaches?
So
Next,
we have
please.
5 main questions. The next one, Raul.
Next slide.
Next slide.
Next slide, please. Yeah.
Next slide.
Yeah. Okay. Thanks.
Hypothesis.
No.
We're on hypothesis.
Yes. Okay. And what is the hypothesis?
Yeah.
The hypothesis is according to the development of the country and the climate. And what can cause the proliferation of antibiotic in wastewater? If we have the wastewater and we have antibiotic resistance bacteria and antibiotic resistance genes, what is the problem? What is the problem if we have the macropollutant? It's good to see that we have bacteria, we have antibiotic resistance bacteria, antibiotic resistance genes, but the presence of a micropollutant has a main impact for the antibiotic, for the transfer genes between the bacteria. But in the developing countries, we have a wastewater treatment plant. Also, we have the community, the waste you will see. We have the animal farming. We have agriculture, and the efficiency of wastewater treatment plant can be considered as a major point for antibiotic resistance bacteria and the resistance genes to reach the aquatic environment and to be accumulated in food chain. Next slide. Yeah. Next slide, please. Yeah. But in developing countries, such as in Congo, that I am working now, the problem of antibiotic and antibiotic resistance bacteria and antibiotic resistance gene proliferation in the non-clinic environment can be considered as alarming for the following reason. Firstly, we have no wastewater treatment plant. We have the hospital if we are discharged into the receiving system without any previous treatment. There is the problem of open defecation. We have no regulation of the prescription and the use of antibiotic in human medicine and the veterinary. You can go to the pharmacy and you will buy without a prescription. And there also, the wastewater is using for irrigation of fresh products. Another? There is a great problem in developing a country, not only in the Congo, but in several. And if we cannot find the solution for that, many efforts that we will do, we will not have a solution. Another slides, please. Yeah. Okay, and if the wastewater reach the receiving system, there is the problem. The accumulation in fish, the accumulation in vegetable, and there also is the approach to detect antibiotic resistance bacteria and the antibiotic resistance genes. In my lab, we developed worldly this approach that we see the bacteria and also the micropollutant, that there is the interrelations and also the coexistence. You cannot only make the analysis or to make the assessment of antibiotic resistance bacteria and antibiotic resistance genes without taking into the account the macropollutant, organic and inorganic macropollutant that have many influence for antibiotic resistance genes and antibiotic resistance bacteria. And there we are working in water, we are working in the sediment, we are working in the fresh products, and we are also working in the fish. And if to take all these matrix and to see what is really the problem for antibiotic resistance genes from the wastewater, and what is the role of temperature according to the country. And after that, after all of the assessment, we can see what can be the solution, what can be the recommendation, and we are doing the GIS, donc geographical system, to make the maps. We make the maps for all of the matrix that we are analyzing. Next. Next slide, please. Okay. Here is that I explained. We have the CLC. What it means? It means that is to see the many generations of antibiotic. We have the last generation of the family of carbapenem and the cephalosporin, that we can see the prevalence of E. coli, we can see the prevalence of Enterococcus, we can see the prevalence of Pseudomonas, this is the opportunic pathogens, and there we can see what can be the multi-resistance. We cannot take only one antibiotic, but we can mix according to the clinical laboratory standard institute that we can make and see for the culture approach and the metagenomic approach. This is very, very important because you can estimate, you can underestimate the antibiotics resistance, or you can upper estimate. And there is a methodological that we take 16 antibiotics and we can mix according to their concentration, according to the species of a bacteria, but we are working mainly with E. coli, with Pseudomonas, and with other enterobacteria. Next, please. Next slides. Yeah. Okay. Here I will give you some examples rapidly. The case of temperate conditions. Here in the Switzerland, you can see that in Switzerland, we have the wastewater treatment plant. Here is in Lausanne, at the Bay of Vidi. And there, the wastewater treatment plant is not efficiency. And we have the accumulation on time of bacteria, antibiotic resistance genes, and antibiotic resistance pathogens. And for that, what is the problem is to know the influence of a temperature, because the temperature of water is about 20 to 25 degrees or under during the summer. And for that, we see the transfer of genes. And if it is accumulated in the sediment, there is the remobilization, but the transfer is less. It is not so much, and according to the dilution. And if we have heavy metals, we have all other organic macropollutant, there is the influence of the pollution for the antibiotic resistance. And we did this, this study for about 15 years to see the influence of WWT wastewater treatment plant for the accumulation of bacteria and antibiotic resistance genes in the Lake Geneva. And then you see well there is the accumulation and the distribution of the bacteria and antibiotic resistance genes. And there in other, you see in the red, we have, if we have many micropollutants, if we have increasing of a temperature, there is also the increasing of antibiotic resistance bacteria and the antibiotic resistance genes. It means that the wastewater is the main and the great source of antibiotic resistant bacteria and the antibiotic resistance genes. And also we see that here we are in the receiving system. And what can be if we can see from all, from some area of Geneva, we can make the sampling of wastewater and see influence of the tourists and other living persons for the proliferation of antibiotic resistance bacteria and antibiotic resistance genes in wastewater. There is the next slide, please. Next slide, yeah. Okay, you see here is the sham data we did from hospital effluent in many units of hospital effluent, and we did the effluents of the communal. We see the communal effluent, and there is a step is wastewater treatment plant, and we see what is the influence of each unit in hospital, and what is the influence of the mixed of wastewater from WWTP wastewater treatment plant and the units of hospital, and what can be the influence for the presence of a bacteria and antibiotic bacteria if we have many sources of wastewater treatment. And it is very difficult to know because there is the mix of wastewater from many units. We did all the units of hospital, and we quantified the OXA, the KPC, the NDM, the VIM, many genes and bacteria. And also we had the treated wastewater, and we see what can be the influence if we have the temperature of about 37 degrees. But it's difficult. And what we did is to go before the wastewater to go to the wastewater treatment, and to make the analysis before treatment and after treatment, and to know the efficiency of a wastewater treatment before effluent going to the receiving system. Next slide, please. Okay, you see here, if you know where the Geneva, you see the quartier of a Paki, quartier of a Bel Air, quartier of Acacia. There you see we took directly the wastewater before wastewater going to the wastewater treatment plant. And this was very good because it was in the period of COVID-19. And there we follow together COVID-19 and antibiotic resistant bacteria, antibiotic resistant genes, and the antibiotics residue. That's very, very good. So the research that there we can see what is the human influence if they are taking the drugs. What is the problem for the water treatment, wastewater before going to wastewater treatment? And we can see what is what can I say? The influence of antibiotic resistance of antibiotics and for the COVID-19. And we follow till now we are receiving the epidemiology of COVID-19 in wastewater. And then we make the correlation with antibiotic resistance bacteria and antibiotic resistance genes. Okay? Another? Other slides? Rapidly. I want to finish. Yeah. Okay, another. This is for the sampling. Yes, another. This is a sampling from the hospital in the units of the hospital that we are doing in Switzerland. We have many units of a different disease that there they are treating the persons with antibiotics. There is no person with antibiotics, and we see if the person they come with antibiotic resistance into the hospital for to see the nosocomial such as MRSA in the hospital units. Another, please. Other slides, yeah. Another, there is for the wastewater, other slides. There is the sampling. Another for the result, yes. Okay, and there you see we are performing many analyses. It's easy. Say, you know, there is wastewater, this is the antibiotic, and to perform the analysis, this is another problem. And we tested the sensibility of many bacteria which can be resistant. We have E. coli, Staphylococcus, we have Pseudomonas, and we have Salmonella, we have Vibrio cholera. And there is that the bacteria that we are working in developed and developing countries in according to the tropical condition. All of the bacteria, we can find it in Switzerland, in France, and other in Congo, Cuba, and that we see what is the horizontal gene transfer between the bacteria. Another slide, please. Next. Okay, this is the result. The result is that ESBL and the CRE are the prevalence in Geneva effluent. There is a great problem. We cannot say that the problem of antibiotic resistance and antibiotic resistance genes are only in the developing countries. You see there is a high prevalence, and we compare this prevalence with the prevalence from other developing countries. But here I will just give you the problem for the Congo, for the situation in Kinshasa in Congo. Please, next. Okay. The situation in Congo, you see, as I explained to you, we have a no. In right, there you see from the wastewater from a hospital without a treatment, and it goes to the receiving system. Here you see in India it's the same, and you see there also there is the toilet near the river, and you see there is the pharmacy. There are many there you can see with many pharmaceutical offices that you can go and you will buy the antibiotic, the drugs without with a medical prescription. And you will see it's a lot of. And from that, we see how to do. And myself, I went with my PhD student, and we bought many antibiotic of this next generation. And there we see that in the river receiving system in Congo, there was many cases of Pseudomonas, and they call multi resistance of the last generation of antibiotic. Next, please. Yeah. And we see that the hospital effluent is not the only source of antibiotic resistance dissemination. There is the wastewater, domestic wastewater. This is another great source because there is the open defecation and there is the no regulation of antibiotic prescription. And the last, next, please. We are in the end. Yeah. Okay. Impacting for the climate. As I explained to you, we compare it. If you see in Congo, in India, in Switzerland, in Congo we have, and India, according to the temperature, we have a high prevalence of antibiotic resistance bacteria in wastewater, in vegetable, and in fish. And there, it's a great problem. You see that the prevalence, for example, for Pseudomonas in India, we have till 60%, in Congo 50%, and in Switzerland. And there is the wastewater can disseminate antibiotic resistance and antibiotic resistance bacteria, but the temperature is the element that is very important. And that we are doing now our research for the influence of a climate change of the high temperature for antibiotic resistance. If we make our research according to the climate, we can tell that climate, to see the climate change, it's also to see the antibiotic resistance genes and the bacteria proliferation in wastewater. That is next. We are in the end. Yes. Next slides. Yeah. Okay. In conclusion, the situation can be considered as alarming in Congo, in India, Cuba, and also now I am working in Morocco. But in Congo, according to our research, untreated wastewater cause a wide spread of antibiotic resistance in form of bacteria, antibiotic resistance genes, and the accumulation not only in water receiving system, but also in food chains, vegetable, fishes, fruits, from the irrigation of the contaminated water to the fresh products. Also, there's untreated wastewater lead to the quality and the degradation of the water body, many antibiotic resistance, and epidemiologic of hydric disease. Because there you will see that there is a resistance for the cholera, for the typhoid fever, and the other epidemic diarrhea. We have the great problem for the resistance in Congo. We correlated between resistance bacteria and the prevalence of the disease. And our study also demonstrated the high infection rate caused by consumption of raw vegetable in salad, donc in high frequency of antibiotic resistance among isolate bacteria. There is alarming situation. And what can we do? What can be the solution for the Congo and other countries? Last. It's the next and the last. Yeah. Next, please. Yeah. Okay. This is the last. The potential solution. There is a difficulty to find the appropriate solution if you have no project. That we are proposing to have the project with ICARS and with a colleague from a University of Kinshasa. There we are working more than 10 years. You can see in my web site, you will see many publications. And here we give some hypotheses to find the solution. Firstly, is to performing with research in different settings. There is no many research performed in Congo. And we have to perform many and to do many researches in different matrix. Also, the acquisition of a quantitative data. If we propose to make the wastewater treatment from a hospital or from houses or from other clinics, I think it's for us, firstly, to have the quantitative data. We have some quantitative data, but easy to see the quantitative data according to the seasonal variation. Also easy to work for the regulation of the use of antibiotics in the therapy, in the clinical and the veterinary. There we did some congress, some seminar, and to disseminate the result from our research to the great public. It was a very small small actions. And there we propose to have a great project to do this, what to do in the clinical and in the veterinary settings. There we propose to collaborate with ICARS and other colleagues. And also as hard to reduce the contaminant from and optimization from a wastewater treatment. But if we have no wastewater treatment plant, what we can do to optimize and to minimize this? This is a great problem that we will do for our future research. Also, we will continue the education and the sensibilization awareness for the public, for the population, for the actor of public health, in the schools. That we will continue with this. And finally, to fight against the climate change, because the climate is the great element for antibiotic resistance, bacteria proliferation spread in wastewater. Thank you very much.
Thank you very
If
much.
you have questions.
I think that was quite very comprehensive and very, very clear presentation. And, you know, we're struck with your conclusion, which is that eating raw vegetables induce antibiotic resistance in people. I think a lot of people, even the medical community, are not aware of that, you know. And when we tell patients, oh, don't take antibiotics like candies, of course we shouldn't. But we have to be aware that every time they eat raw vegetables, they're at risk in poor countries. And also, I remember what you told me once in Geneva, which is that even in Switzerland, and it's true also in France and the US, when there's heavy rain and flooding, and we see a lot of flooding internationally in rich countries, we had it in Spain recently, we've had it even in Dubai in April. We have a lot of epidemic related to flood. And of course, flood means wastewater treatment are not equipped for these climate change problems of wild weather. You want to make a comment on that? Did you hear me? I think you didn't hear me. John?
Yes, I can't hear you. Yeah.
Oh, I had stated that, two things, that the eating raw vegetables is a cause of antimicrobial resistance, as you outlined it, to the extent that the vegetables are irrigated with contaminated water. We had that crisis even in the US, where we had to go without lettuce in supermarkets for 6 months, if people remember that one. And it's often forgotten in giving this hand-me-down advice on people in poor countries don't take antibiotics like candy. Of course, we shouldn't take antibiotics like candy, but we have to make sure that the food supply is safe. And I wanted to ask you a question about the fact that whenever there's flooding, and there's been flooding in Dubai in April, we've had flooding in regions in Florida and elsewhere in the US, we've had floodings in Spain recently. Whenever there's flooding, even in well-to-do, relatively well-equipped country like Switzerland, France, or Spain, or the US, when there is flooding, there is wastewater, some wastewater is being released into the clean water. And this, of course, is a source of AMR. Can you comment on that?
No, it's
flooding.
Yes, it's a problem for that because for us, it's firstly to assess the antibiotic resistance in wastewater and in water using for irrigation. This is the first. Secondly, we take the irrigated plant and then we analyze the antibiotic resistance bacteria and antibiotic resistance genes in the plant. Donc, it's many the raw vegetable. Also, there is the problem that we are doing is to see the transfer of the gene under the vegetable. And to see also the vegetable coming from the developing countries, from Congo, from India, from Thailand, and to come in Switzerland. And there we are doing this aspect, and we are proposed a monitoring, how to make the monitoring of there's a plant, and how, firstly, secondly, donc how to see if we can make to set up the problem of to treat the water before using it to the irrigations. For the last solution is if we can make the treatment of the water before using it to the irrigation of a plant products. Now we are doing in the tank, we are doing the experimentation with moringa, one plant that we call moringa for the tropical conditions, and we can make this moringa to treat the irrigation wastewater before doing it in the raw vegetable for irrigation. Yeah.
Thank you very much, Professor Pote. That was a brilliant presentation. We have to go to a last intervention, which is taken by Luther King Abia Akebe. Can you come online, or do you want us to try to play your pre-registered presentation again?
I am online. I had a question there that talked about, I had a question for the previous presenter where I mentioned, I was asking, he talked about the accumulation of this bacteria in the aquatic system and the sediments. I just wanted to know if they conducted any studies that could tell how long those organisms could stay inside there.
Yes, it depends to the physico-chemical parameters of sediment and wastewater. First is the organic matter. Secondly is the temperature and the other biotic conditions. Because we found in the accumulation, in the sediment accumulated, that more than 100 years from the datation. And from the sediment, we found that there are antibiotic resistance genes active in the sediment. The sediment can be a reservoir of accumulation. It can be, donc the bacteria can be killed, but antibiotic resistance genes are released. As aquatic ecosystem is the hotspot of a transfer genes, the genes can be transferred also again to the active bacteria. And there is very difficult to calculate. I can say to you that this is the permanent action. The solution is to limit the source of input to the aquatic environment. For the bacteria such as E. coli can be for 3 or 4 weeks if we have no many organic matter. But if the E. coli is resistant, there will be the bacteria will not be active, but the genes will be out of the bacteria cell and to be accumulated to another bacteria. And it's such as a cycle. Okay, but in the sediment, if it's accumulated, it can be accumulated more than 1,000 years of persistence of antibiotic resistance genes. It can be degraded, but you have the gene sequences, the sequence of the genes, active genes that are in the sediment. And if they are near the bacteria, there can be that we call horizontal gene transfer that can be performed by three mechanisms such as transduction, conjugation, or transformation. That the problem of antibiotic resistance bacteria and the resistance genes in aquatic environment is permanent.
Okay, thank you very much. Thank you very much. You'll be changing the slides for me that side.
Yeah.
Thank you. And once more, Garance, thank you and the production team. I see there's been a lot of technical hitches, but at least we are still going and we are still learning. Previous speakers have spoken a lot about antimicrobial resistance surveillance. They've already spoken about wastewater treatment plants. We've spoken about One Health. So in this little presentation and brief presentation, I'm going to be talking about wastewater surveillance of AMR in South Africa. Although I'm mentioning South Africa, it will be more or less the African continent. And the question I will be posing here is, are we building a castle in the air, or is it really a practical solution which we can use as an early warning for AMR within the continent? So we can proceed with the slides, please. Next one. So I decided to look at the whole point of looking at what is wastewater first, because when we look at other studies and many studies that have been published, and they say they have tested wastewater, and some people have reported on wastewater epidemiology, the definition of wastewater has been a little bit different, and what people have defined as wastewater has been maybe in their own context. And when we look at wastewater-based epidemiology, it therefore brings us to understand what wastewater is, so that we know where are we sampling and what are we reporting in terms of epidemiological data. So in this case, I look at wastewater. If we look just at the basic definition of the Oxford Learner's Dictionary, we say it is water that contains waste substances, just as basic as it is. It could be from homes, from factories, from farms, or any place, as long as it contains waste substances. Now, if we come down to the household, which is domestic, we look at gray water, again, which is water that ideally has not had any contact with fecal matter. And we have the black water that has contact with fecal matter, or the kitchen sink, because it has food substances in there and other animal waste. But if we also see, we see that gray water falls within the same category. The kitchen sink water will fall, other than kitchen sink. So that is it. And then when we look at the industrial wastewater, we look at wastewater that comes from an industry, irrespective of whatever thing is coming from there, as long as it is used for any of the processes within the industry. But in this case also, when we know that industries are not isolated entities, and within industries, we also have sanitary facilities, what do we therefore say? Is the industrial wastewater, is it gray water, or is it black water? As we will realize, most of our systems are all combined, such that the gray water from the industry together with the black water from the industry will be combined into one system that will be channeled into the same sewer channel pipe into the main treatment system. Next slide, please. So, having said that, all those water that we have mentioned in systems that are sewered, that are connected to a central sewer system, which you will agree with me is not the case in many low and middle income countries, especially those located in Africa. Those that are connected to a wastewater treatment plant, we therefore regard as the wastewater treatment plant as that facility that will clean that water, whether be it gray water, be it storm water, be it industrial water, be it whatever water it is. That system should be able to clean this water, to remove the stains or the waste inside to acceptable limits. That is one thing I want us to understand, to acceptable limits, either for discharge back into the environment or for use for other purposes. Next slide, please. So this therefore brings us now to wastewater-based epidemiology. Now, with wastewater-based epidemiology, this is just an approach whereby please, can you off your mics on the other side? I'm getting your voices this height and I'm being so with wastewater-based epidemiology, therefore, what is happening is that it is quite challenging to go into the community. If we take in the case of diseases, for example, or AMR, it is quite challenging to go into a community and to sample every single individual, or even if we take a representation of the community, it becomes challenging to sample every individual in that sample, in that group that we have taken, or the cohort that we have taken, and to come out with epidemiological data. So instead of doing that, we say, okay, if we combine, if this community is connected to a sewage system, then it means that what is coming from the household will come to the same system. What is coming from the hospital will come to the same system. What is coming through stormwater and the farms will come into the same system. So sampling at that one system is that we have created some sort of a microcosm that contains everything, a whole mixture of everything that is in the community. Just like having to collect individual samples and pull them together and then to analyze. So by this approach, it therefore becomes easier to tell what is within the community. If we were to look at hospital wastewater alone, for example, we might be getting AMR that is representative of the hospital. Not everybody goes to the hospital. If we look at AMR within the community, we might miss the one that is coming from the farms. If we go just to the farms, then we might be missing what is coming from the hospital and the community. But if we look at a wastewater treatment plant that is a centralized system, then we are looking at a combination of all of this. And while we would think that wastewater-based epidemiology is something that is new, if we look at the timeline down there, it was already used since in the 1940s. So it is not a new concept. It is an old concept. It's just that with the advent of COVID-19, for example, its importance became became more apparent, because using that approach, it was easy to tell the burden of COVID in regions where it wasn't possible to test everybody. Next slide, please. Okay, so as I was saying, we have conducted several studies over the years on analysis. I wouldn't put it as wastewater-based epidemiology per se, but we've conducted several studies on wastewater treatment plants in South Africa under the directorship of Prof. Abia, as Garance you were mentioning earlier. And this is just one of the studies that we conducted. And in this case, we were looking at what was distributed in a certain community in terms of extended spectrum beta-lactamases, lactamase producing E. coli strains. And please continue to the slide. And what we realize is that when the isolates that we got, both in the influent and the effluent, had diverse percentages of resistance to all the antibiotics that we tested, just as you can see on the table. Please continue. If we also look at the variants of the beta-lactamase variants, they will see that there were also different variances depending on where the sample was collected. Please continue. The next slide is of greater interest to me. Please continue with the slides. Next slide. So now, when we look at this, we decided to do a clonality of the isolates that we got. If we focus just on the influence, because in the influence will be more representative of what was within our community. I just want to mention here that we sampled 6 wastewater treatment plants within the same district, so that we could have a coverage of the district, because it is serviced by 8 wastewater treatment plants, 2 of which are not functional. So the 6 that were functional, that was receiving waste, we sampled those ones. So if we just look at the influence and we look at the number of clusters or the number of clades that we have in our phylogenetic tree, we'll see that there is a diverse spread of extended spectrum beta-lactamase producing E. coli within that community. So by just looking at this graph, we can tell that it is not just one strain that can be circulating within the community, but it's very different. And if we look at the effluent, we see that looking at the ESBLs, we see that it's more than in the influent, indicating that the wastewater treatment plant could have engineered or could have favored the acquisition of this resistance within within the treatment, and this is the water that goes back into the community, because this was the effluent of that's the final effluent that was going directly into the river. Next slide, please. So we conducted several other studies, as I mentioned, but what I want to point is that the importance of using this wastewater treatment plant allowed us first to, when we sequence our isolates, we picked up a novel isolate, right? That of a novel Klebsiella isolate that was not reported before. And we see that from there, we can say that it's possible that we see a new strength not previously reported within the community. The same as we reported that we had the first report of the colistin-resistant genes within. So the wastewater-based epidemiology, therefore, in this case, this approach helps us to understand that, look, while we might have people going to the hospital, while we might sample or study people, it's very possible that we miss some of these traits that are coming into the community. And if we use the approach properly, then it's possible that we can detect these traits quickly and then to inform the health sector and to see if measures need to be taken for such monitoring to be done within the community, and maybe call upon the hospital and to start looking for such traits. Continue, please. So when we still look at South Africa, therefore, we conducted a review in 2022 to look at what was done within South Africa, therefore, in terms of wastewater surveillance. And when we look at this map, this shows the distribution or the various provinces of the country. And the first, the A there represents the population of the country. The B is the number of wastewater treatment plants that within that community, within that province that service the population. And then the C is the number of wastewater treatment studies that have been conducted and have reported on AMR in wastewater. Again, as Garance had earlier mentioned, Abia was doing a lot of work. That is why you will see that KwaZulu-Natal is one of the places where you had the highest number of studies. So KwaZulu-Natal had 14 studies together with some other provinces, but most of the provinces have below 5 or don't even have studies at all, meaning that these studies are not yet well distributed within the country on a national level. Next slide, please. And also, remember that I started with the definition of wastewater. When we look at the type of wastewater that was reported in the studies, although wastewater treatment plants formed the bulk of the studies that were reported, other studies reported domestic household, domestic wastewater, for example, without sampling the wastewater treatment plants. Some other people reported hospital wastewater, meaning that the studies that were reported were not all based on wastewater treatment plants, which is that whole microcosm that receives the hospital and all other components of the community. So basing on one of the study, let's say if we base just on household or just on hospital, it's possible that we miss other aspects of the resistance that is within that community. And if we look on the table below, looking at the first column, we'll see the organisms, which I'll come back again. If we look at the media of isolation, we'll realize again that although some studies used check for the same organism, the approach in which they isolated this organism or they detected the organisms were completely different. And I just did not put everything, but even the number of sampling at times that was conducted, at times some studies sampled just once. Some studies had longitudinal studies where they sampled every month. Some studies were sampling every 2 weeks. Next slide, please. So this is just the representation, therefore, of the organisms that were reported in the studies within the time that we reviewed. And you see that E. coli is the most studied organism. And while some other studies went deep into looking at specific pathogens, but most of the studies reported either E. coli and Enterococcus, obviously because those are indicator organisms. Please proceed. So what therefore do we identify as a gap? Although I'm mentioning this gap now, if we look at the spatial or geographical gaps in the study based on the first map that I showed, we see that the wastewater-based epidemiology studies within the country are underrepresented, meaning that only very few provinces are representing the data of the country in that sense. And this is the same principle in the whole African continent and most low and middle income countries. And this spatial gap at times is because although we have all those provinces serviced by or connected to wastewater treatment plants, some of these wastewater treatment plants are not functional. If we look at the current green drop report of South Africa has a green drop report which is produced every year that evaluates the state of most of the wastewater or all the wastewater treatment plants, functional wastewater treatment plants. We realize that about more than 50% of the wastewater treatment plants were functioning below the actual or the effective functioning capacity, meaning that it's either they were just not working completely, or when they were working suboptimally, meaning that they don't treat enough sewage before sending them inside the thing. So this already brings in this spatial gap where if the wastewater treatment plant is not working, it's not receiving any waste, then it's not possible to sample in that particular place. So we cannot talk about wastewater-based epidemiology in this area, in this case, because there is no wastewater system which we can sample. If we look at the methodological gaps, the studies, some of them did typically phenotypic studies where samples were collected, analyzed through culture, and exposed to antimicrobial resistance to antibiotics, and also even the number of antibiotics and the various types of antibiotics to which the isolates were exposed were different in all the studies. Some people will test 5 antibiotics, some will test up to 20 antibiotics. There is no consensus onto what antibiotics need to be tested. Some of the antibiotics were not even supposed to be tested in the first place. You know, like it's just like getting enterococcus and testing them with gentamicin 10 milligrams, for example, which already we know that we need to test them for high level resistance to aminoglycosides. And this therefore presents a challenge in that we cannot compare the studies within the various provinces because the approaches that they use are completely different. Some other methods now, some other studies did entirely just metagenomics, whereby although they report resistance, they don't say whether these organisms were viable but non-culturable. They don't say whether these organisms were even alive in the first place. Maybe it's just DNA that was present inside it. This also presents another gap because we also saw that some of them could not conduct these studies because they didn't have the facility. While some were doing genomic studies up to whole genome studies, like the KZN group, for example, some other provinces don't even have the facility to do that. And again, it's expensive for them to do. Microorganisms gap, we see just from the table that not every study was looking for the same organism. If we want to compare across again, we cannot compare the level of resistance within that. And then in terms of reporting gap definitely applies to all the other ones, just like what is reported. Some people focus their reporting just on the organism of interest, but organisms that were not of interest obviously will not be reported. The same as genomic studies will report, for example, just on the genes, but will not talk about the culturability of the organisms. So there is that reporting gap becomes different, and it becomes challenging again, as I mentioned, to compare the studies in each of these provinces. Next slide, please. So what therefore do we take out of this study? We realize that if wastewater-based epidemiology is going to really work in South Africa and the African continent, then we need to standardize the protocols for assessing the wastewater. We need to identify what is wastewater. We need to define what the wastewater is and where we need to sample. We need to consider the sampling regime. Is it once a month? Is it once every month? Once every week? How many times do we just do a point prevalence, collect one sample, go back, and then make people run up and down? Or because if we do a once-off sampling, for example, a hit does not necessarily mean that there is a problem. Neither does a miss necessarily say there is no problem, because on the following day there could be a pollution event. There is the need to build capacity in terms of sequencing technologies. It is gradually improving from the time we published this article. It's gradually improving, but bioinformatics skills are still very lacking. Sequencing is still very expensive for most of our universities, talk less of other low and middle income countries. Funding must also be made available to researchers, as the sequencing technologies are expensive, as I have already explained, and the reporting of works on AMR needs to be improved. We need to create some sort of a repository that will inform us, or to which other researchers that want to conduct their work can easily go and say, you know what, there is no need saying this, or what they have seen is what has been reported somewhere else, or what they have seen is novel. If we don't have a centralized system, if we don't have a proper database, then it becomes difficult to know whether what we are seeing is a cause for concern or not. Next slide, please. So I would like to conclude with this slide. When I was mentioning that, I'm talking about South Africa, but if we look at the low and middle income countries, and we look at the percentage of treatment, water that is treated, waste that is treated on our key, we see that more than half of the African continent, for example, does not treat any of the water at all. There is no data on that. And only few countries like South Africa, Botswana, and some other ones treat up to 50% of their waste. So all these other countries are either not connected to a wastewater treatment plant, or the few wastewater treatment plants that are existing there are not even functional. And that brings us therefore to the question to say, how can we therefore carry out wastewater-based epidemiology in low and middle income countries, especially African countries, if we do not have wastewater treatment plants at all? Do we go to individual households and sample the wastewater that comes from there? Do we go in the informal settlements, for example, in South Africa, or the slums of Kenya? Do we go there and collect the water that is running off the surface and analyze that to know what is within the community? Or do we keep on just preaching and saying that we need to carry out wastewater-based epidemiology because that is the future of epidemiology? And are we not building a castle in the air in these countries when the facilities to, first of all, even test is not there? So that is why I was asking, is it a practical solution, therefore, for African countries? Is it a castle in the air? Based on that, the discussion will be open, and then maybe we should need to look into which methods will be appropriate to discuss or to approach wastewater-based epidemiology within low and middle income countries, South Africa, and Africa as a whole. Next slide, please. So I acknowledge the following people that took part in the work. I've not mentioned everybody, but the funding organizations are there. So thank you very much for your kind attentions. Your questions can come in. I will be very happy to take them up. Okay, I see there is a question here. How does the Prof see the national wastewater treatment system's future in South Africa after the last general elections? It's okay. The nice thing I like to talk about this question is the fact I'm not going to link it directly to the elections or whatever thing is happening. But one thing I want to say is I was so pleased to see that in the last UNGA meeting, the issue of AMR has been politicized. The biggest challenge in bringing AMR to the politicians is that it has no political thing. Like, they don't see what can be used out of that to motivate some political campaign, you know. But now that it has been politicized, where governments have been called upon to introduce it fully in the system, it now becomes a whole thing of national concern, such that every person that we want to talk about now will probably tell you, look, AMR is a problem, wastewater treatment plants are a problem, so I am going to build wastewater treatment plants. If you vote for me, I'm just saying, then I'm going to ensure that the country does not have wastewater treatment plants that are not functional or pipes that are bursting. And you see, the community starts to understand, because now there is a political agenda that is attached to it. So I believe that South Africa is making good progress. Like in the animal sector, for example, antibiotics are gradually moving away from animal production, and they have advised the companies, the pharmaceutical companies, to stop writing on antibiotics that they are promoters. And those that have, some people have voluntarily agreed to remove. Those that have refused to remove, they have been refused the license to market the antibiotics within the country. So there is progress. And as I mentioned earlier, it is also in the National Action Plan, which is being revised every year. And just recently, the Department of Health is also working on a technical working group that will ensure that AMR in the environmental components is being taken care of. The next question is, did you see the impact of antibiotic-resistant bacteria and algae in food chains from South Africa? Yes, we did. Yes, we did. In my previous presentation, I mentioned that we used the farm-to-fork approach proposed by the Register Protocol. We used the farm-to-fork approach to study poultry from the moment they hatch until the time they were packaged for the shop, like from the farm to the table. We did the same thing with the pigs, and we saw that there was antibiotic resistance across all the continuum. That is from the feces to the trucks to the litter that was produced onto the meat portions that were being marketed. So we found AMR in that, and this obviously, although it wasn't that high in some cases, but it's still a cause of for concern. And as I'm saying, it's a gradual move away from the use of antibiotics within the country on animal farming. And we hope that with that, there is going to be a change. I know we can't just eliminate it entirely because biosecurity in itself, it's not very cheap. But at the same time, the farmers need to be told, need to be given an alternative. For example, we just can't tell them, okay, don't use antibiotics anymore. How do I know that one chicken in my flock of 1,000 is sick? So I might as well just give them all. But if I am giving an alternative, I am giving a way I can go about to prevent my birds from being sick, then I have greater chances of not using it. And also, what is the gain? If I stop it, what do I gain? If I don't stop it, what do I lose? You know, that is all what has to get into the discourse. But truly, we saw antibiotic resistance and resistant genes within the food chains. And we have a couple of studies that have been published on that from our previous students. I can always share the papers with you via email.
I'll say something. Thank you very
Yes.
much, Professor Abeke. This was really a really delightful, very instructive presentation. I would like to conclude this webinar with one comment. I would like it to dedicate it to Jim O'Neill, because 10 years ago, he initiated the AMR review when he was still Secretary of the Treasury under Cameron in the UK government, you know. And it was quite remarkable if you look back at Chapter 6, because Chapter 6 with the London School of Economics looked at what would happen to antibiotic consumption if we had WASH, in other words, safe water and wastewater in India, Brazil, Nigeria, with 4 countries. And they concluded that it would reduce antibiotic consumptions by 60% and save several millions of children's lives every year. And that, you know, that was 10 years ago, and it's sort of being forgotten because we still have to talk about it today. So if I resume what you said and what you said Pote before, it was just tremendous. I mean, we look at 80% of the antibiotics are used in agriculture. Those you know, figures have been used everywhere. And of course, without proper wastewater treatment in low and middle income countries, which is a good part of the world population, it means that we are releasing genes of antibiotic resistance into the water supply, into the soil. The bacteria exchange them. It goes back into the vegetables and the fruits. And of course, in the advanced sector, we import green beans from Kenya and so on and so forth. So if you look at it, it's really a One Health global trade and, as you said, a political problem. And we are not going to make any dent by just telling well-to-do countries' patients to behave and take their antibiotics properly, because that's not the crux of the problem. The crux of the problem is inequality and lack of real industrial development in poor countries. And until we do that, we're not going to do any progress whatsoever in AMR with the new pill. And I can quote my friend Dominique Monet, who is lead AMR for the European Centers for Disease Control, who says, just putting a new antibiotic on the market in the present situation, and he was talking about Europe, will be just like putting fuel on the fire. We need to have we need to have clean healthcare system, even in Western Europe, in order to be able to make a dent. And today we have conflicts springing up everywhere on the planet. And the fact is, conflicts, metals, waste do produce antibiotic resistance. So I thank everybody. I think it was very successful. We had some technical problems, but that's life. And I would propose that we go towards doing a publication of what everybody said, because it's a very comprehensive approach. And I invite everybody to join us again the day after tomorrow on Wednesday, midday, for a section on airborne with Tim Walsh, who is lead AMR for Oxford, who also worked in China and is now working in 6 countries on newborn antibiotic resistance. He is very well known internationally, and we have with us 2 of the most important, most qualified world experts from Hong Kong on airborne transmission, what it means, and how we can slow it down. I thank everybody. Again, I think it was a remarkable presentations. Thank you very much. See you on Wednesday. Bye.
Bye-bye.